All right, we're going to go ahead and get started. Good morning and good afternoon, everyone. Welcome to the Global One Health Initiative April webinar sponsored by the National Institutes of Health Fogarty programs. Thank you for joining us today, especially considering the circumstances. We have two themes today, including wildlife diseases as well as COVID-19 in animals. And we will be hearing from Dr. Risa Pesapane, who holds a joint appointment in the Ohio State University College of Veterinary Medicine, as well as the School of Natural Resources, and Dr. Jeanette Quinn from the Ohio State University College of Veterinary Medicine. And with that, I will hand it over to you, Dr. Pesapane. Please go ahead and share your screen whenever you're ready.
Okay. Okay. Hello, everyone, or Dumela, as they say in Setswana. Thank you to GOHi for inviting me to present today. I am a disease ecologist and professor of ecosystem health at Ohio State. But because I'm new here, the work I'm going to talk with you about today actually occurred at my former university. So let's get started. Throughout this talk, it's really important that you try to envision the landscape of Chobe and keep that image of this ecosystem in the back of your mind. So Chobe is in the northernmost corner of Botswana. Let me turn on my laser pointer here. So we're up here. And the area down here you can see is all Kalahari. So this region is very arid with very little surface water. And the study site is nestled right up here along where these four countries meet at the Chobe River, nicknamed the Four Corners of Africa. And the Chobe is an extremely important source of permanent water for the hundreds of thousands of animals in these countries during this dry season. And it results in some of the densest and most stunning wildlife viewing in the world. So of course, what does that mean? It means that there's a lot of ecotourism. So I wanted to zoom in and show you a little bit about what the town of Kasane looks like. So it's a very small town, entirely supported by the ecosystem industry. It's really there to support that industry, to be fair. And everything to the left here is all national park. This is the airport here. And so you can see the town really follows the river for a short period of time. So everything or everything in this town here is there because it's very close to the entrance of the national park, and this national park is borderless, so it has no fenced border, which means that wildlife can freely roam out into human environments. So this is obviously really wonderful for the wildlife, but you can imagine that it does create some conflict with the humans. So along the Chobe, we have these really elaborate, stunning, world-class 5-star lodges juxtaposed with the local villages that support this industry, which most of the buildings look like this. Of course, some of them also don't have finished windows and may or may not have running water. But there is a lot of wildlife, as I said, that enters town. So just like in the US, we might have turkeys or deer, fox, and raccoons that mill about our town. Similarly, in Kasane, they have warthogs, baboons, mongoose, and elephants that interact with people fairly regularly. And of course, outside of the site of the lodges, you may get dumping into wildlife areas. So in Chobe, we have a human population that is affected by malaria, leptospirosis, diarrheal diseases, and some of the highest rates of HIV/TB in the world. And we also have a wildlife population that has pathogens like canine distemper and rabies and some other emerging pathogens. And many of these species here are endangered or threatened. So the question is there potential for microorganism exchange? Because we have these two vulnerable populations and there's potentially negative outcomes for both if spillover is happening between them. So the thrust of this research that I'm going to talk about today is not necessarily focusing on the health of one group or the other, but it's really to study whether there is sufficient connectivity between them to facilitate microbial exchange. And to do that, we really need an ecological model that includes a sentinel species and a model organism. So I chose banded mongoose as sentinels. These mongoose are fossorial, so they den in the ground and forage for insects and invertebrates. They are happy to range between human and wild environments. They love using man-made structures like drainage culverts for denning. They're really attracted to this lush, artificially watered ground. So as you can see here, this is Chobe Game Lodge that is right along the river, and they of course do this really extravagant landscaping, which requires a lot of watering to maintain grass. And you can see that it's mostly desert outside of that. So for a species that likes to forage along soft soils to look for invertebrates, this is an ideal place to hang out. They are also opportunistic in the sense that they're willing to eat garbage, and they've been seen licking cooking pots and drink containers that belong to people. These mongoose are also highly social, so they form troops of 4 to 60 individuals, and they engage in a lot of social grooming and care. But more importantly, they aren't just social with each other. They often forage alongside, or in the case of this short video, they forage on neighboring wildlife. They can also be consumed by predators as well as people. So mongoose are really good sentinels for the health of humans because of their exposure to our waste and the built environment. Plus, they're very good sentinels for wildlife because they regularly have intense interactions with other species. And therefore, they can act as like a conduit between the built and the wild environments. Importantly, banded mongoose are the host of an emerging wildlife disease in the MTB complex, and it's a novel TB. It's concerning because it's actually transmitted cutaneously. It's related to human TB, and so there is concern that Mycobacterium mungi could spread to other wildlife or perhaps even people. So as with most emerging infectious diseases, M. mungi is difficult to study because we have very few developed methods for detecting it. Transmission events are likely very rare. So instead, if we want to know whether there's sufficient connectivity to permit this spillover, we need to use this model organism. And the organism that I chose was E. coli because of the well-developed assays we have to differentiate populations of E. coli, because it's been prolifically used in other microbial exchange studies, and it's the most common indicator of human microbial pollution. So beyond knowing if microbial exchange is happening at all, it's really valuable to understand what pathways that might exist for this exchange. So other studies have found, shared microbes between park rangers and wildlife, which was pretty groundbreaking, but they didn't investigate the mechanisms by which this exchange might happen. And in the Chobe area, there's actually little direct contact between humans and wildlife. There is some infrequent consumption of bushmeat, but the vast majority of contact between humans and wildlife actually happens indirectly through their shared environment. So if you remember, Kasane is a small town, but it experiences these massive influxes of people during peak tourist season. And when that happens, that's usually during the dry season, because that's when all the wildlife are coming to the river. So when that happens, the sewage system often can't keep up with this influx. It wasn't built for these periods of high use. It was built for more of the typical use. So what we see is we get pipeline breaks, we get overflowing drains. And of course, this is at the same time that there are hundreds of thousands of animals flocking to the Chobe River, literally 300,000 elephants. They're eating, drinking, defecating on the riverbanks. And year round outside the lodges, there are also practically very few clean toilets. So much of the local population practices bush latrining. So what we end up with is sort of a constant low-lying pulse of human and animal waste, coupled with these intense periods of fecal loading in the environment by humans and animals in the dry season. And the result is widespread contamination of a shared resource like soil and water with those fecal microorganisms. And if we directly sample human waste in the environment, then we can make conclusions about shared microbes and at least one mechanism of exchange. So taking a step back, if we want to really simplify this system, here's what we hypothesize the system to look like. We have humans that are sharing microbes with wildlife through environmental contamination, and wildlife feces coupled with sewage is then contaminating a shared resource, which is the Chobe River. And ultimately, this could come full circle back to humans, since they do fish, bathe, and even drink in the water. So the project I'm going to talk about today is actually focusing on this right side of the system. And at the end, if we have time, I'm happy to tell you about the follow-up studies that were on the left side of this system. So to determine whether humans were sharing microbes with wildlife through fecal contamination of the environment, we collected isolates of our model organism, which was E. coli, from sources of environmental waste and our sentinel species, the banded mongoose. And we subjected these isolates to a 3-tiered study design that included antibiotic susceptibility testing, box PCR, and MLS TPCR. So the reason we chose to do antibiotic susceptibility testing is because E. coli actually occurs in many animals and environments, and it was important that we first confirm that an isolate had originated in the human body so that we could then properly conclude that it came from humans into the environment, into wildlife. So we did the susceptibility testing on all of our wildlife samples, and I looked at resistance to 10 commonly used antibiotics that span 6 classes, antibiotic classes, and these were all available to people in the local hospital. And then to determine the similarity of E. coli between humans and wildlife, I used an efficient method of DNA fingerprinting called RET-PCR that amplifies the short non-coding regions and generates a barcode for each isolate. And then we can create a phylogenetic tree of these isolates to see if they form one population or if they demonstrate some sort of population structure that would differentiate human isolates from mongoose isolates. And lastly, to further confirm whether that phylogenetic relationship observed using DNA fingerprinting is appropriate, I took a subset of these isolates and sequenced 7 housekeeping genes. And this step not only provides confidence in our RET-PCR relationship, but it can also give you some insight into how recently spillover occurred. So mongoose sampling collection is laid out in this map. So again, here is the Chobe River in blue. And black dots here are going to indicate troops that had known clinical cases of Mycobacterium mungi, and the white dots are troops that did not have any positive individuals. And we sampled 6 of these troops in all. So we sampled 3 of them, which is within the national park in pink here. So here's our green national park. And we also sampled 3 in the village areas. And the stars indicate lodges. So you can see that 5 of our troops are associated with a lodge, and all 5 of those were positive for M. mungi, as are many of these troops. But we also sampled 1 troop that was in the park but not associated with a lodge, and that happened to be a troop that was negative. So we obtained E. coli isolates from mongoose by non-invasive sampling. The mongoose conveniently practice something called social latrining behavior every morning. They all exit the den together and they each defecate immediately. So because we had radio collared a few members of each troop, we were able to locate them in their dens early in the morning, right before they emerged. We wait there and then we'd immediately collect the freshest species we could find. And we ended up achieving fairly balanced samples between, inside the park and inside the village. And then we cultured E. coli from these species and confirmed that it was in fact E. coli by PCR. And to represent the human population and environmental contamination, I sampled from bush latrine sites, sewage overflows, and the local sewage evaporation ponds. So this is all of the waste from the town that has flush toilets. This, of course, would represent people who are not using flush toilets, and then breakage, actual seepage into the environment. So we ended up with 43 of these samples. And I did also collect a few samples of soil that was not contaminated as sewage as a somewhat of a baseline, basically to determine whether or not there was just E. coli everywhere, and that that would tell us that this sewage seepage in the environment was an irrelevant factor. So we wanted to make sure we did test that too. So here's a breakdown of our samples. You can see here, so these are our different troops. Again, the pink is in the park and the yellow is in town. The fecal sample numbers from different troops varied, and this really reflects largely the variation in the size of the troop. Like I said, they can range from 40 to 60 individuals. For example, Cephalana here was a really small troop. At a previous census, it only had 6 individuals, so we were pretty happy that we were able to get 8 samples from them. It probably included everyone in that troop. But basically, important takeaway here is our total number of samples, which we collected over 200, including 46 of those coming from the environment or from representing humans. And we're able to culture E. coli successfully from over half of those. And then the isolates went through antimicrobial resistance testing, rep PCR, and EMA list T PCR as follows. So some results. Our antibiotic testing revealed surprisingly widespread resistance among mongoose troops. So every single troop, all 6, demonstrated individuals with at least some level of resistance, even within the national park. And more than half of the mongoose, so 57%, harbored isolates that demonstrated resistance to at least 1 antibiotic. And before I move on, I want to point out that HP, it's called Hippo Pool, this is our troop inside the park that is not associated with any lodges. And what was interesting about this one is that even though a large number of the isolates or individuals had isolates that were resistant, they were largely only resistant to ampicillin. I think there was only 1 other isolate that was resistant to anything else. So we suspect that this actually reflects probably naturally acquired resistance to ampicillin, which has been described in literature before. So in terms of multi-drug resistance, a full quarter of our mongoose harbored isolates that were resistant to 3 or more antibiotics that we tested. The troop with the most multi-drug resistance is CGL, which is Chobe Game Lodge. And again, this is actually within our national park. This group had not only the highest percentage of resistance among isolates overall, but it also hosted the most multi-drug resistance. And so 1 isolate from this troop actually was resistant to 9 out of 10 of the antibiotics we tested, which was very concerning. We know that this troop in particular prefers to den in the soakaway of the septic tank that services the employee accommodations at Chobe Game Lodge, and that those employees also sometimes will throw them raw meat scraps from the commercial kitchen. So as for the overall profile of resistance among mongoose isolates, we see that ampicillin is most commonly followed by resistance to, resistance to ampicillin is most commonly followed by resistance to our tetracycline class. And again, you see that overall resistance in our park troop, CGL, which is in brown here, or brownish red, is again just remarkably high. When we look at the rep PCR tree to see if the E. coli were similar, we actually find that E. coli from humans, sewage, and mongoose all comprise one population. So I know this is a big tree on the right here, but essentially I've color-coded the different things here to be mongoose or human sources. And what's important is that none of these clades are specific to mongoose, people, or sewage. Like they're all completely intermixed, and we have a single monophyletic clade of isolates here. So when we run our population genetic analysis, we learned two things. First, we see that genetic diversity among our various E. coli strains is high within a population, but it is, is higher within populations than it is between our populations. And that second, we get an FST of nearly 0. That's here. And if you're not familiar with what FST tells you, basically it ranges from 0 to 1, and 0 being completely identical and 1 being totally different. So this confirms that there is no significant difference between these populations, between the human and mongoose E. coli populations. And in fact, they're quite nearly identical. So next, I took an isolate that some isolates that represented each of those clades in the former phylogram, and I performed this MLST PCR to see if the relationships held true based on sequence, and indeed they do. We see that mongoose isolates fall out together and along with humans in one monophyletic tree with very few genetic differences between them. So these two types of molecular approaches agree. So all of this resistance in mongoose was really alarming, and I really wanted to feel confident that the antimicrobial resistance was coming from the human population, because we weren't directly swabbing people in this study. We wanted to be sure that the fecal environmental contamination that we were finding in our sewage samples was representative of the human population. So we partnered with the local primary hospital, who generously offered to share the results of all of their resistance testing in clinically isolated microorganisms. So for the drugs we tested in common, I could see if the patterns of AMR in humans matched those from sewage samples and also mongoose. That was very special. And here are those results. You can see that actually the resistance profiles mirror quite well. My human fecal waste samples show resistance levels very similar to what is found at the hospital, and so do the mongoose, however, at lower prevalence levels. So what did we learn? We learned that humans are actively sharing microbes with mongoose and Chobe, which means spillover either from humans to wildlife or potentially from wildlife to humans is possible. We found considerable resistance to antibiotics, regardless of whether animals were in supposed protected areas. This is obviously concerning from a medical standpoint, because we know that bacteria can exchange resistance genes, and we know from the literature that this is even more likely to occur among gut microbiota. We confirmed that fecal contamination of the environment is a potential pathway for microbial exchange and a definite source of antimicrobial resistance. And this means that we have evidence of a recurring mechanism for spillover that can happen between humans and wildlife. And because it's contaminating a shared resource, this, that humans also use, this is a huge concern. So lastly, we now know that since mongoose have this propensity for environments that are contaminated with human waste, and they do become infected with our E. coli that is resistant, they may spread that resistance across the landscape as they traverse in their home ranges, defecate, interact with other wildlife, or interact with human structures. So I think I have time to share what happened next. So the question was, are mongoose actually representative of all local wildlife, or are they just special? So coming full circle to this idea of fecal contamination in the environment as a connection between humans and wildlife, there were some additional studies that examined the Chobe River as a common source. So we conducted additional sampling along the Chobe River and looked at feces that were deposited by different species, as well as collected samples of suspended water samples for E. coli isolation from the water. So in a nutshell, in one slide, if I can tell you the story of what ended up happening with the follow-up studies, is that when they looked at 18 additional wildlife species, they found that mongoose are not special. 43% of isolates from wildlife were resistant to antimicrobials. And fascinatingly, that AMR and MDR were influenced by the trophic level of these species, their life history strategy, like whether they were water associated or terrestrial, and whether or not they had a tolerance for human environments, which the take home lesson from that was that if we monitor apex predators and water or human associated species, that might be most informative for AMR pollution in the environment that could affect us. They also looked at a 30-year data set of diarrheal disease reports from health facilities and compared that with climatic variables, and found that rainfall is driving diarrheal incidence during the wet season in Chobe. And this lends more evidence to this idea that shedding by infected individuals into the ecosystem, flushing that into the waterways, may contribute to subsequent diarrheal cases. So again, this is more evidence that the Chobe River is a common resource connection. And lastly, that antimicrobial resistance in E. coli that was isolated from the river directly itself, we found that it was widespread in the water. And it was highest in wet season and along urban areas. And again, that AMR profile was nearly identical among humans, animals, and the water in Chobe. So we have this really beautiful picture of this connected ecology in Chobe, and again, more evidence that environmental contamination of antimicrobial resistance by humans initially is impacting wildlife, and then all of this wildlife is also inputting fecal matter into the soil, which then sits during the dry season. There's no rain, and then you have this huge rainfall, which mobilizes everything in the soil, and it runs into the river, and then the river becomes contaminated, and then of course people and animals use the river. So I just wanted to include this in case people wanted to follow up. These are the studies I just mentioned, and that's where I'm going to end. So I hope that you agree that Chobe is an absolutely magical place to visit. I hope you agree it's a great model for interdependent human and animal health. And the work that I presented to you today was conducted at Virginia Tech. It was supported by the National Science Foundation, the Wild Eyes Foundation, and the Morris Animal Foundation. So thank you.
Thank you, Dr. Pesapane, for that very, very important and highly relevant presentation. We do have time for a couple questions. If you could raise your hand in the sidebar, that would be excellent, and we can take maybe one or two. Or you can send a message in chat as well. All right. Excellent. Okay, we have a question from Dr. Gabrias. Go ahead.
All right, thank you so much. Risa, great presentation, great work, and very interesting work indeed, considering the wildlife-human interface, but also phenotyping, genotyping parts, which makes it more powerful. So I just have a quick question in terms of the antimicrobial susceptibility findings you have, which is predominantly ampicillin. Of course, there are various MDR, Enterobacteriaceae, MDR occur in various ecosystems. Was that your expectation? But mainly also, what is there any information in the antimicrobial selective pressure? That's my first question in the environment or in the ecosystem generally. To what extent do you have that information? And secondly, with ampicillin, you think it's intrinsic, but since you did a PCR, is this beta-lactamase based or is this efflux pump based gene? Can you elaborate a little bit? Thank you.
Thank you, Dr. Gabrias. So actually for our antimicrobial resistance testing, we did the Kirby-Bauer disc diffusion method. So we were looking at streaked lawns on a plate. So I don't have molecular answers to the mechanisms for resistance for those, but I can tell you that in that area of Chobe, there is almost no veterinary care, and there is also no farming. There's very little subsistence farming, so there's not a lot of use of antimicrobials except for in the human population. So likely relatively low selective pressure in that sense.
Yeah, great, thank you.
All right, excellent. So we're actually going to have to move on. But if you have further questions, I can forward them to Dr. Pesapane. And if it's all right with Dr. Pesapane, include your email in the follow-up webinar email in case anyone has any questions.
Sorry, Laura, Risa, I think there is another person's question I see on the chat that what is a policy implication? Somebody asked on policy implication. Can you comment?
Okay.
I can say that based on these reports, we definitely were speaking with the Ministry of Wildlife and the local community and talking about the importance of containing garbage and containing human waste so that there was less access by wildlife. And we also reported to them what we were finding with the sewage and how much of a problem it was. And hopefully this could result in changing how they treat water there, because also the E. coli and other fecal particles can adhere to sediment and things, which change with wet and dry seasons. So it's very different than what areas that don't have that very polarizing season experience in terms of cleansing their water. So that's also something we talked about, but largely it means that there needs to be some infrastructure changes because they're not, they don't have the capacity to manage that amount of human input at certain times of the year. So that's a much larger expense and would require more government funding to make that, to improve that.
Great, thank you.
All right, thank you very much. And we'll go ahead and move on. I'd like to introduce Dr. Jeanette Quinn presenting on COVID-19 in animals. You may begin.
Thank you.
Ready.
Thanks. Can you hear me okay?
Yes.
Okay, excellent. And you can see my screen?
Yes, love it.
Wonderful. Excellent. Thank you very much. And thanks for inviting me to talk about this. COVID-19 has kind of absorbed my last couple of weeks as we've been very busy responding to everything from the College of Veterinary Medicine, changing our entire curriculums and working with animal shelters to put out a lot of guidance documents, also working with CDC, AVMA, and others. So I wanted to share kind of an overview of what we know or what we think we know, which may very soon be what we thought we know, but we've learned something new. So almost as soon as you put something together, it seems to become outdated. So I'm just going to try to give a broad overview and hopefully have time at the end for any questions that you might have. I wanted to start with this graph because some of you may have seen it in its original form, shared mostly by researchers, veterinarians, public health professionals, epidemiologists, showing the whole flattening the curve concept. And then somebody turned it into cats and everybody shared it. So apparently that's the way to to reach your audience, is to turn things into cats. But this really does highlight the issue with having a whole lot of cases really fast in a naive population, whether it's animals or humans, versus trying to slow down that infection rate so that we don't overwhelm our resources. Oh. All right. My slide is not advancing very well. Let's see what I can do about that. There we go. So no, that went too far. Okay, so I just wanted to share a little bit of a timeline. I promised that I am going to focus mostly on animals, but this is a human disease, so I think it bears discussing a little bit. And I'm also just absolutely blown away by how rapidly this has spread, as I'm sure many of you are. So the first cases around December 1st in Wuhan, China, spread locally for a little while there, and they reported to WHO at the end of that month, right at the end of the year. And things picked up pretty quickly after that. In January, they identified what virus it was, and that it was not SARS, but it was a very similar virus. They had the first fatality, first cases outside of the country of China, the first case in the US. And by the end of January, there were nearly 10,000 cases in 21 countries, and WHO declared a global health emergency. February and March, we're seeing it expanding very quickly to other areas, increasing in numbers. Italy and Iran had really severe outbreaks, and then towards the end of that period, OSU started increasing their cases logarithmically. And of course, all of the mitigation efforts that have been put in place, I'm sure you're all familiar and living with a lot of those restrictions to try and reduce those cases. So it has had an effect, and I'll show you a little graph in a minute to kind of highlight that. But if we look at where we end up at the end of February, 82,000 cases, the end of March, March 31st, over 300,000 cases. And then just 2 weeks later, over 2 million, I'm sorry, in the US, March 31st, 300,000 cases. So what I put in here was showing how we were doubling about every 2 days at the end of March just in the US. And then of course, globally, over 2 million is where that's the landmark that we passed yesterday with a very high number of deaths as well. So this is definitely spreading. Here's a heat map, kind of shows total confirmed cases. We know that there's a lot more unconfirmed, and then the number of deaths associated with that. If you're not familiar with this dashboard, it is a really great source for up-to-date information. They update it several times a day, and I'm happy to share that link if anybody would like that. Here's the graph that I alluded to. So Ohio has actually gained quite a bit of attention. And I wanted to brag just a little bit about that because I'm pretty proud of our colleagues here who've, I think, really provided good advice, and to our government who has accepted that advice. So this graph is one from a model that our IDI group has been working on. In yellow is what they expected if we hadn't done anything, if we hadn't canceled schools and churches and eliminated group meetings over 10 and social distancing, shelter in place, all of those things. If we hadn't done anything, that was expected, with a peak of about 62,000 cases in one day. And every day they update this model with the new data that's come in. So this was sort of in March. This was what they were predicting. We're kind of here still on the upside a little bit. This is what they were predicting our flattened curve was going to look like based on the data that they had so far. And then just a week ago, they ran it again, and you can see how much more that curve has been flattened than we even expected. So it's really nice to see that, and we're hoping that we can maintain those low levels. All right, I shared that because I really wanted to highlight, as we talk about animals, that this is a human disease. There are 7 coronaviruses that infect humans, and you can see the 4 on the left here. They're just human to human. There's no animal involvement at all, and they cause the common cold, essentially, very, very commonly seen, cause cold symptoms. And then we have now 3 other coronaviruses here, SARS, MERS, and COVID-19. And they all originated from bats and most likely traveled through an intermediate host. So we have the civet and the camel, and we're not sure about what the COVID came through. So there's been a lot of speculation, and there've been some papers. So one paper came out a couple weeks ago that proposed that snakes were the intermediary. That one was torn apart, I guess would be a good way to put it, by the rest of the scientific community. And so that's not believed to be actually what happened. And then just a couple days ago, adding a lot of sleepless time for me, they published a paper that said that stray dogs were the intermediary host. And very similarly, that's been, I wouldn't say disproven, but certainly called into question. It's not believed to be a well researched or well supported argument. So the most likely candidate right now is the pangolin. And if you're like me, I did not know what that was. So here's a picture. It's sort of a scaled anteater. That still is open. They're still looking into that. So expect more, probably many more theories on that before we find out. So this is a great chart that AVMA put together showing some of the common coronaviruses in animals. And there are quite a few. So dogs and cats each have their own. Swine have more than their fair share. I think there are 5. Cattle have a couple. I'm sure that veterinarians and other healthcare providers on here have treated these various diseases. The majority of them are enteric, causing gastrointestinal issues, and a few of them are respiratory, like we're seeing with COVID right now. And a few of them have vaccines. I wanted to mention, because I get this question a lot, there's not expected to be any cross protection between these vaccines and COVID. So there's no reason to vaccinate animals hoping to protect them from COVID. And there's definitely no reason to vaccinate people hoping to protect them from COVID. These vaccines will not do that. They're very specific to the virus that they were designed for. And these are very host specific viruses. So we don't see them spilling over into other species or into people. But as people are now fearing the term coronavirus, when they start to hear it in animals, and again, very common, dog and cat especially, and to have those viruses, they may not understand and may assume that we're talking about COVID. So always good to keep that in mind when we're talking with our clients. So lots and lots of stuff in the news. I'm trying to put together some educational things for students, and I wanted to do some hot topics in the news, and I haven't been able to find any hot topics. Everybody's talking about COVID. And a lot of concern, worry, and really a demand for more information, especially surrounding pets, because pets play such an important role in our lives. For many of us, you know, they're living in our houses, they're sharing our bedrooms and sleeping in our beds and on our couches. And so there's arguably a good reason for them to want to know, right? So every time something new comes out, there's a whole new flurry of articles that go around. So I wanted to go through with you guys what we know, what's been published and what's happened so far that can help us sort of inform decisions and have a better picture of what's going on. So with dogs, we'll start there. There were 2 unrelated dogs from different households in Hong Kong. Both of them lived with a positive owner who spent a lot of time with those dogs, and they were both PCR positive on respiratory samples. They were quarantined for a period of time and tested serially. Neither dog had clinical signs of disease, and both of them had a weak positive. They were unable to culture live virus from the Pomeranian, but they did culture live virus from the German Shepherd. and they both did seroconvert. So these are believed to be mild infections, but actual infections that occurred. They also have tested at least 25 other dogs that were known to be exposed that were all negative. For cats, we have one positive cat in Belgium, PCR positive on fecal and gastric fluid. The cat had both respiratory signs and gastrointestinal signs with diarrhea and vomiting. So that's very interesting. We have limited information, so we don't know if there were other tests done or if the cat had some other illness. And this was an incidental finding, or whether the PCR, or sorry, whether COVID actually caused those clinical signs. Antibody tests are pending. So we haven't seen, I'm sure they've done them, but we haven't seen any reports out of there yet. There was one cat in Hong Kong, unrelated to the dogs, that was PCR positive. No clinical signs for this cat. And in Hong Kong, they tested at least 14 other exposed cats, and all of them tested negative. All right, so far we're really looking at 2 dogs and 2 cats. And then you probably all heard about the tiger. This got a lot more attention in the media. In the Bronx Zoo in New York, a tiger tested positive on PCR. It's a 4-year-old Malayan tiger who had a respiratory illness, and it was progressing. It was not severe, but it was progressing, and they wanted to run some tests. So they actually kind of decided to test for COVID just as a lark, actually. They didn't expect to find it at all. They said, why not? We're testing for everything else. Let's test for that. That decision actually has received a lot of criticism because tests are in such short supply worldwide for humans, and especially in the United States, where many people who need to get tested aren't getting tested. So it's probably good for me to highlight here, while we don't recommend unnecessary testing of animals, these tests were not competing for human test kits. They were not human test kits at all. They were run at animal labs using different reagents and different things. So just thought I'd highlight that because that's come up quite a bit. There were 3 other tigers and 3 lions that also had respiratory illness, very similar signs. They were not tested. They weren't severe enough. They really wanted to test the one as a representative sample. So we don't know if they had COVID or not. The zoo's been closed to the public for a while. So while they don't know exactly how this tiger was exposed, or the others if they had it as well, they do presume that one of the keepers was contagious, either asymptomatic or pre-symptomatic, because they were checking all of their staff. Nobody was febrile, nobody had clinical signs. All of them did get better, by the way. So all of the tigers and lions recovered without any additional need for treatment, just ran its course. And same with the 2 dogs and the 2 cats. The first dog did pass away, but it was unrelated. He had some pre-existing health conditions, being a 17-year-old Pomeranian. There's a few serosurveys that have been done that I thought were interesting to share as well. So in Wuhan, China, of course, where this started, they did some serology. They did 39 banked samples from cats from before the pandemic, and those were all, I'm sorry, I put that they were all positive, but that is incorrect. They were all negative. So all of those pre-samples were negative. That's my fault. 15 out of 102 were positive during and sort of, I guess, following, if you can say we're there yet, the outbreak. The titers were very low, except for 3 cats who all lived with one owner who was sick. And all of them were negative for PCR at the time that they were tested, which isn't surprising if they were already seroconverted. They tested 9 cats and 12 dogs in France. This was a group of veterinary students. 2 of them were confirmed to be ill. The rest had compatible illness, but weren't tested because of the shortage of tests. So they were presumed positive. All of these animals were negative on PCR and on serology. Okay, so that's kind of all we have with naturally acquired infections or exposures. There's two studies that are experimental that I wanted to share with you because I think these have gotten a lot of attention, and it's important to kind of understand how they fit into the rest of the information that we have. So the first one was published last week in Science, and they looked at ferrets, cats, dogs, pigs, chickens, and ducks. So they did pretty similar things to all of them. They took 5 puppies and inoculated them with incredibly high doses of virus, far, far higher than you would ever expect from a natural exposure. And then they took 2 puppies that they did not inoculate, and they housed them all together. So 2 of the 5 inoculated dogs were PCR positive for a very short period of time, and no live virus was recovered. And those 2 seroconverted. The other 3 that were inoculated did not. And the 2 dogs that were not inoculated but were co-housed with those that were inoculated tested negative on all tests. And none of them developed any clinical signs of illness whatsoever. So from that, they're concluding that the dogs seem to be pretty resistant, that they may get a mild infection with no evidence of transmission. They did the same thing with cats, except for this time they inoculated the 5 cats. They housed 3 individually in cages, and then next to each cage, they housed another cat that was not inoculated. So they wanted to see if that indirect contact could spread. So they weren't actually in the same cage, but they were in adjoining cages. So they did see, they also euthanized a couple of these cats several days in so that they could look to see if there were lesions and also to see if they could capture live virus associated with that pathology. And they did see lesions pretty much isolated to the respiratory tract, but quite prolific around there from tonsils, upper and lower respiratory tract. They did have positive PCRs on fecal samples, even though they had no lesions in the GI tract. And those 3 cats seroconverted. One of the 3 that were not inoculated, but were housed adjacent to inoculated cats, did develop not clinical signs, but did test positive, and seroconverted. So we did have transmission from the inoculated animals to a naive cat that had not been inoculated. They repeated this with another set of cats that were younger kittens, and they found that the pathology that they showed was more severe compared to the older kittens. So this is showing that the cats are susceptible to infection when they're exposed, and they do develop pathology from that. And they do shed at least low levels of virus. So with the ferrets were the ones that they really reacted. They were sick, they had fever, they weren't eating well, they had pathologic lesions associated with their clinical signs, PCR positive, live virus everywhere, and seroconversion. And they concluded from their work here that ferrets could be a suitable model for studying COVID in people. So I imagine that we'll see a lot more research on ferrets in attempts to understand this disease better. The good news is pigs, chickens, and ducks were also inoculated, but none of them tested positive and none of them seroconverted. So they seem to be, at least from this small study, pretty resistant. The second experimental study was published in Cell Host and Microbe, and they looked at ferrets alone. Again, they were looking to see if they could use them as a model as they used them in SARS. And their data really supports the first study. They did have illness, they had positive tests, they had live virus, and they had ferrets who had not been inoculated getting the disease from ferrets who had been inoculated. So they were transmitting in that environment. Again, remembering that these are really, really high doses initially. And I think that's something we have to keep in mind. The experimental studies are telling us what's possible. but we need to look at natural infections to tell us what's probable. And when we start to look at that, we have 5 cases, and we have over 2 million human cases. So clearly this is not a significant issue. It's not something that we should need to worry about getting disease from animals or having them be involved in our transmission cycle. Clearly this is a human disease, and there are a few cases with either high doses of exposure, close contact with their owners who are sick that can get infected from that. And again, all of the naturally infected cases were very mild, and we did not believe that there was opportunity for transmission there. Another important point is we really shouldn't be surprised by this. A lot of people reacted like this meant the virus was getting worse, that something was happening that was, you know, scary and bad, when really it was just us learning more about the virus. It's not surprising when you look at what we know about similar viruses. SARS spilled over to cats and to ferrets and to pigs. Previously, not in high numbers, but we did know that those were possible. And this virus is very, very similar to SARS. Early on in January, an article published in the Journal of Virology showed that the protein on COVID binds to the ACE2 cell receptor, and that cell receptor is very prominent in humans, but also found in cats, ferrets, and pigs, and of course some non-human primates. So just knowing that, we could expect that in those species, we would have the risk of infection. And it seems to be that our studies and our natural infections are supporting that for cats and ferrets. But the pigs in the one study did not seem to be susceptible. So again, those are all small studies, so hopefully additional work will be done in those areas. We don't have any testing that I've seen published yet on other livestock species, but it's not expected to be a significant concern for infection. Okay. So I think I already summarized this just before this slide. So we don't have any evidence to support transmission from animals to humans. They're just not part of the transmission cycle. And if you have a pet that gets sick in the household, you know, you have to realize that the person who exposed that pet was the larger, greater risk for the other people in the household. So just keeping things in context all the time. Livestock and food safety, I think this is an important aspect. Even though we're not seeing disease, I think livestock and food safety are definitely impacted, mostly because of the impact on people. So many things are shut down. Restaurants, some stores, butcher shops, things like that are shut down. And so the demand for meat, milk, and produce is dramatically decreased. And farmers are dumping milk. Farmers are destroying crops. They have animals that they're sometimes euthanizing because they can't take them to slaughter and sell them, and it's just costing them money to continue to provide care for them. So there are some impacts there that I don't think are fully realized or fully understood. Another challenge that we've been facing is transportation disruptions. So just moving food and produce around from location to location, or animals even, has been a challenge. I think everything is starting to kind of smooth out a little bit, and some of those things are opening up and not being as much of a problem. But at least in the United States, and I imagine in other countries, we're seeing that. I do not see any changes in regulations on import or export, again, further supporting the fact that there's really not concern for horses or livestock to be involved in the spread. Another question that I get asked a lot is about animals as fomites, or more correctly, mechanical vectors. So if you sneeze on your pet and you're infected and the virus lands on them, right? Is there a risk that people could contact that just like they would contact other surfaces in their environment? And that's a really difficult one to answer. There aren't studies that look at that directly. We have some studies that look at survivability. In general, it's not believed to be very long-lived. on fur coats or fabrics. It's believed that that virus would dry out pretty quickly and die off because of that. Also, if it settles into the coat, the chances that you're going to contact it by petting them isn't really high either. So CDC and others are saying that it's really no significant risk whatsoever. Other public health agencies in Canada and other places are like, yeah, we don't think it's going to be a problem, but it's always good to wash your hands and to take basic precautions. And I would second that. It's very simple to mitigate that potential risk. It is a low risk, but it's very easy to mitigate that through hand washing, and just if you're sick, avoiding giving that virus to your pets, right? So if you are sick, what do we want to do? Treat your pet like you would any other member of your family, right? So if you had your kid there, you wouldn't curl up on the couch and cuddle with them and then sneeze and cough on them. You would keep your distance from them. So do the same thing with your pets, and that will help reduce any exposure that they might have to potentially develop an infection or to get the virus on their coat. Again, risks from those are incredibly low, but it's not zero. So we do want to be cognizant of that. You can have someone else provide basic care for your pet. If you have to be the one to provide care, maybe you live alone, you're going to want to wear a mask, wash your hands before you handle your pet, and then after you handle your pet. Again, avoid coughing and sneezing on them, cover that to reduce contamination of the environment. Discard your tissues in a location where the pet can't get into that, especially if they're one to go through the trash can. You want to make sure they can't get into that. Those are not good toys for your pets. And this is really important, to make a plan to have someone else care for your pet if you need to go to the hospital, for example, and you're not able to care for your pet. You don't want to just leave them without anybody to care for them. What happens then is the animal shelter will come out and pick them up, and they will do that, and they are providing that service, but really trying to reduce that. And the best place for a pet is in the home, and the next best place is in the home of a friend or a family member. So just trying to keep that so that they're not having to be brought into the shelter. All right, am I running out of time here? I am. All right. Let me see, I'm trying to bring up my chat so I can see what you guys are saying. I can wrap up here and take some questions, if that works, or I have just a couple more slides I can present. That's up to you.
I would suggest to go ahead, Jeanette. This is interesting.
Okay, great. I think it is too. So that's probably why I'm talking about it so much. So how do we care for our pets when we're sheltering in place, or we're on quarantine, or we're self-isolated because of an illness or an exposure, or because of the government order? Right? If we stay home, our pets should stay home too. I've gotten a lot of questions from people who take their pets to visit nursing homes and do other therapy type things. And while those are really valuable for the people involved, it's not worth the risk of spreading this disease. So pretty much anytime you say, can I do this with my pet? I would say, would you do that with a person? And then that will help you decide. So if you're staying home, they should be staying home with you. And you may not be able to go out except for things that you absolutely need. So that means when you're planning for what you need, plan for what your pet needs as well. You want at least 2 weeks of food, favorite snacks, toys. One of my friends is posting frantically for her cat's favorite toys. She can't find it anywhere online. Make sure you have any medications that they need. Again, at least 2 weeks worth. Veterinarians can call in prescriptions to a human pharmacy. So if your veterinary practice is closed, they're still probably doing telemedicine and doing prescriptions and things like that. So you can still reach out to them for that. And then just treat them like any other member of your family. Really restock those supplies when you go out. It's a good idea to have a go kit too, where you have like everything that they need in one place in case something does happen and somebody needs to come over and take your pet with them for a period of time, everything will be in one location. Medical records, vaccination records, things like that are great to have that in that emergency kit. And we stress so much that there's no need to give up your pet due to concerns about your animal getting sick from you or spreading disease. We feared in shelters that we would see a lot of people dropping off their animals, especially when the news reports started coming out. And I have to say, we haven't seen that. There's been a little bit, but not nearly as much. And we're very excited about that. In fact, adoptions are up. So people are staying home. Seems like a perfect time to welcome a new pet into their home. So adoptions are at record highs across the country. And they also expanded foster programs, right? So we don't want an extended stay in the shelter for months. That's a really hard thing to handle mentally for an animal to be confined for that period of time. And especially when we want to reduce the staff responsibilities so that they're not having to come out and put themselves in danger. Reducing the number of animals in the shelter was paramount. So recommended to reduce intake to only what was essential for public safety and animal welfare. So if somebody just wanted to rehome their animal, they're being asked to keep it for now and delay that, or to use other means besides the shelter to find a new home for that animal. Also, increase in adoptions and increase in foster care. So foster is, if you're not familiar with that, is a sort of a temporary home away from home. So it's kind of like a vacation, I guess, from the shelter. And then that person can decide to adopt, or they can bring it back to the shelter for adoption at a later point. We use it a lot for sick animals that need care, that don't need to receive that care in the shelter. So that frees up resources in the shelter and gives that animal a home environment to recover in. So this is sort of a novel approach to use it, you know, as a way to just get these animals out of the shelter. And it's been phenomenal to see this, so many shelters posting empty cages all over the place. And there we go. Another question that we get a lot, can I walk my dog during quarantine? So we talked so much about everybody staying in. They weren't sure if they could walk their dog. So absolutely, it's good exercise for you. It's good mental stimulation for your dog. Take them out for a walk. Just be really careful about how you do that. Go somewhere where you don't expect people to be. Choose a time when it's not busy. If everybody goes out and takes a walk at 6 p.m. in your neighborhood, then maybe go a different time, and make sure you maintain your social distance as well. This is something that I was very, very happy to see, as governments were very quickly trying to decide what workers were essential, what services were essential. All of them that I saw declared veterinarians, farmers, animal shelter staff, other animal care providers, especially those involved in food production, to be essential. Was really glad that we didn't have to do much in the way of lobbying for that to happen. Now, that doesn't mean that all of the activities are essential. So elective surgeries, booster vaccines in pets, that might vary for livestock if that's going to be an immediate disease concern, but for most of our pets, if they're well vaccinated, they can wait a couple months for a booster. So those types of things are being put off. Lots of creative work being done in shelters and in veterinary clinics with curbside pickup. So you have an appointment, someone comes out with PPE and picks up your pet, and then the owner talks with the veterinarian or the shelter staff on the phone or over Zoom like we are to gather all that information and discuss options, so that there's not that, you know, standing in the same room discussing those kinds of things. And there's a lot of increase in telemedicine going on as well. I already mentioned these things with shelters. Another benefit for the sheltering community, aside from obviously having these animals in a better environment for the long-term stay and reducing the staff that are needed to come in and care for them. Some shelters have closed completely if they don't have public services. But those that are providing public services are seeing an increase in pets being brought in from homes where their owners have gone to the hospital or have passed away, and those animals have nobody else to care for them. So all of the earlier efforts have been really helpful in freeing up space so that they're able to bring those animals in and quarantine them and then get them ready for a later adoption. So just general precautions. I probably don't need to highlight this. I think we're all very, very familiar with the physical and social distancing. Fomites are really important to keep an eye on. It's not a very common method of transmission. Mostly it's close contact, droplet, but the fomites can move this virus around. People move this virus around, and the things that we take with us move this virus around. And so we need to be very cognizant of that, and we need to be cleaning and disinfecting all the time. The good news is that pretty much every soap and disinfectant is going to be effective. This is an enveloped virus. It's very easy to kill. Right. Survival data, I thought this was really interesting. I wanted to share. This is the only study. I would like to see more because these results are always so, they're so closely tied to the environment that they were conducted in as far as temperature, humidity, and all those kinds of things. But they're looking at about 3 days of survival time up to on non-porous surfaces like plastic and stainless steel, which fortunately are very easy to disinfect. Porous surfaces like paper, cardboard, fabrics, things like that, less than a day. And the droplets, when we cough and sneeze, can kind of hang around in the air for about 3 hours before they start to settle out. And then the smaller particles can hang around longer, but they're not as common, not as frequent. All right, and so with that, thank you for your patience while I wrapped that up. I'm happy to take questions.
Well, thank you, Dr. Quinn, so much for this very interesting and again, highly relevant presentation. We can take several questions, but first I would like to note that Dr. Jeanette Ali, director of our GOHi regional office, will be giving a talk on a recent survey work that GOHi has carried out with COVID-19 in Ethiopia this coming Saturday, April 18th from 10 to 11 a.m. Eastern Standard Time. I will include this information in my follow-up email and also send it in the chat box. And Dr. Jeanette actually has a question for Dr. Quinn. So Dr. Jeanette, please go ahead.
Thank you very much. Thanks, Dr. Quinn. Wonderful presentation and highly relevant. I want to take this opportunity of you knowing the African setup and the way, you know, pets and dog ownership and free roaming dogs and other animals are being handled, and considering how people may react if I mean they see that we are having infected dogs or whatever. So I wanted to know from your experience reading, and you also told us that those who are already confirmed are from either Europe, Asia, or the US, and I wanted to hear your recommendation to the veterinarians, public health professionals, and even the community living here in order to detect early if at all animals are infected. We haven't seen any report from Africa yet, and also how to manage the case. And so far, all the education and public health communication, risk communication is coming, assuming that is, again, you also emphasize it's a human disease. So I want your professional advice, knowing the local situation in Africa, to the policymakers, professionals, and community. What kind of advice do you give over?
So facts, not fear, I think is going to be one of the most important things. And I would strongly recommend that those who are going to be putting out messages get together and talk about what they want those messages to be and come up with some talking points, and everybody shares the same talking points. We did that with our sheltering community, which is pretty diverse in the United States, lots of different types of shelters, different groups that they don't always see eye to eye. But I was really impressed with how well everybody came together, identified concerns, and then developed evidence-based solutions and talking points, and shared those. And it has gone really well. When the media reports things from a balanced perspective, because you've informed them and you give them those talking points, then we get a better response. As far as what the risk is, I really do think it's like, I think almost every case that happens in an animal is like worth writing up. I mean, it's just that rare. We may see more in cats, I'm not sure, but it does seem to require pretty close and prolonged contact. So when I think about how animals are living in Ethiopia, especially the free roaming dogs and the free roaming cats, I think the risk there is even lower, right? I think that I would have a very, very low level of concern, but a little bit if it was someone who, a pet who was living with someone who was sick and it was like always on their lap and they're coughing right on them, things like that. And even those cases that we've seen where that type of thing has happened, they've been very mild or not clinical at all, and recover just fine without any other treatments. So as far as managing those cases, if you suspect them, I don't think you need to do anything special. I would if, let's say you had a dog or a cat living in a home with a person who was sick and the animal gets sick, I would just continue life as normal. That person's quarantined or isolated, that animal's isolated with them, and they'll recover on the same schedule, and then they'll progress on the same schedule. So I don't know if that answers your question. We did have a case actually that I just learned about in the US with a woman who had 14 cats in her house, and she was sick with COVID. She was positive, and one of the cats developed respiratory distress and went to the ER, and then 4 other cats acutely developed respiratory signs. And so they did test them, and all of them were negative for COVID. So it's important to keep in mind that there are a lot of common diseases for dogs and cats that cause respiratory infections, and they're far more likely than COVID is. So I don't know, did I answer your question?
Yeah, pretty much. Thanks.
Yeah, great. It's a good question. It's great to hear your voice.
Same here.
Excellent. So we'll go ahead and take 1 or 2 more questions, and then I will forward the rest to Dr. O'Quinn via email, if that's all right with you, Dr. O'Quinn.
Sure.
So one question from Abebe Balete says, thank you, Jeanette, for the time-rounded data you shared. My question is, 1, so far experiments had been conducted around dogs, cats, ferrets, birds, and pigs, and the conclusions but for us Eastern Africans, and particularly Ethiopia, we have different parks hosting many wildlife, and our dependency in contact with livestock species like cattle, goats, and equine is very high. So what will be your advice for us for, one moment, it just cut off. So what will be your advice for us for the ahead of it prevention and control of COVID-19? That's the first question. If you want, okay, and the second one, do you think we professionals in animal health have the readiness to control COVID-19 if it occurs as an epizootic or panzootic state, keeping in mind that it will be a dual burden as vets, as frontliners, as it is occurring in humans?
Those are both really good questions. I hope that they do some studies with some more species to see, even though when they do a challenge study and they give them these really high doses, it's not necessarily indicative of what will happen with a natural exposure, but it does tell us what might be possible. The pig study showed, and I'd like to see that repeated, that they were not able to induce an infection even with really high doses. They found, I believe, that to be true for SARS with some of the other livestock species. So I would expect that they would find the same thing with with COVID, with the livestock species. So while we don't have proof that they can't become infected, it doesn't look likely that they would become infected. So I think that's really good news. And I think that with as many human cases as we have, that we would probably be seeing it if there was going to be an issue. So I'm starting to breathe a little bit easier just because of that. Are we ready? That's another question. And this is a good exercise, right? To say, what if? What would you do if? Because that would certainly change the landscape of this disease. I don't think we're ready in the US. So I don't think Ethiopia is ready. I think that we both have a good start, and we'd have to figure it out as we went along what that response would look like should that occur. But I'm very glad that I don't think that's a reality. I think that planning for that would be a good academic exercise.
Excellent, thank you. Well, for the sake of time, I think we'll go ahead and wrap up, and I can forward other questions to Dr. O'Quinn.
Sure.
So yeah, thank you again for these wonderful presentations. And we'll go ahead and close. Everybody stay safe, and thank you for joining us today. And yeah, we'll see you next time.
Yeah,
Bye.
thanks. Bye-bye.
Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Thank you so much, Jeanette, and everyone. Thank you. For those on OHeart program, I see Professor William O'Gara as well. Is this the same line or a different, Laura? I think it would be great for NIH D43 OHeart fellows. Dr. Zelalem is coordinating the meeting. Either, I don't know if it's the same link or a different one. Laura, please alert us, or Zelalem, and we'll continue now. Yeah.
I think